To begin our lab, we started by preparing our meter, in which we used two buffers to do so, one with a pH of 4 and another with a pH of 7. After following the meter prep directions, we were able to successfully calibrate our meter. We then tested the pH of common solutions to gain practice recording data and to have a better understanding of the different levels of pH. Weak versus strong acid. Strong acids have a higher concentration of hydrogen ions and will completely dissociate in water, making their dissociation equal to 1, while weak acids only partially dissociate. Because of strong acids' small number of ions, a faster reaction occurs to reach equilibrium, as well as a greater change in pH. On the other hand, weak acids have a larger number of ions, which means a slower reaction occurs to reach equilibrium with a smaller change in pH. In order to minimize the change that acids have on a pH, a buffer may be used which will either bind or release hydrogen ions to maintain a particular pH. In order to continue with our lab, we needed to prepare a buffer solution. A buffer solution is a solution that resists this change in pH when limited amounts of acids or bases are added. It contains a weak acid and its conjugate base, or a weak base and its conjugate acid. A buffer maintains relatively constant because the buffer contains both a weak acid that can react with added base and a weak base that can react with added acid. In addition, the acid and base components of a buffer solution must not react with each other. After preparing our buffer solution, we were able to record its pH. We recorded all of our pH results into a data table. Lastly, we properly cleaned and put up our pH meter and all other material used, leaving our lab area clean.